1977, that's another technology, DNA sequence. So I think in some of my genetic lecture, I like to ask, okay, in 1977, there are two scientists independently developed a sequence method. You probably only learn one in the class, but in your genetic class, we'll talk about Gilbert's method as well. But this is another example. This is molecular biology. Yes, this is molecular biology, but this technique made the data available quickly. We used to have only the protein sequence because we are very good in chemistry. So we get to know proteins, and we know proteins is important. We know the sequence of the protein, and we know DNA much later, right? We recognize DNA as an important molecule in life. It's a very later thing. It is 50 or more years later than what we know. We recognize proteins. This technique let us get to know more about this DNA sequence, and we get the data quickly. And this is an important part. And of course, later the technology becomes even better because we can do it quickly. So this is the method and provide the amount of data. Then in 1981, so this is the scientist used the crop-cross DNA to try to reconstruct phylogeny. So see, the technology developed and people start looking into our cell. And there are two organelles. It's important, right, in plants. And we only have one. We only have mitochondria. But in plants, they have chloroplast and mitochondria. They have their own DNA. And it's relatively abundant. So we can get it relatively easy. So you can see all the things happening because the scientist is ready, the technology is ready, then the data is ready, then the method. So everything happened in similar type of order. So every breakthrough, there's conceptually, data-wise, and methodology. So this is like people are thinking about using the DNA information from organelles to get information. So this is a phylogenetic reconstruction. We're not particularly interested in one specific gene on the molecule. We are interested in how this DNA sequences can tell us the history of those organisms. And similarly, we can use different technology. So in 80s, there's a lot of different things happening at the same time. So DNA hybridization is another thing. Well, probably it's very hard to understand. If I can get a base pair data, I mean, I can get A, T, C, G. It looks much clearer about the information and why I want to do DNA hybridization. Maybe you can switch the view, ask yourself, why? A lot of people are doing genome sequence. There's so many genome centers. People are doing genome sequence one after another. After the human, we have a lot of other things. So why? So this is like you want to see things from a sample point, or you want to observe an object, have a global view. You all learned, we have a story about how blind people knows about the elephants, right? All the Chinese history, elephant is rare because elephant doesn't really exist in mainland China or the mainland of China. So all the elephants, so the story always starts from in time, there's another kingdom send a gift to the emperor of China. And the gift is an elephant. And the story follows. And everybody look at the elephant because this is the first time people know elephant, and it's a huge animal, and it's really special. However, if people cannot see, they don't know what exactly elephant looks like, so they touch it. So that's the sample point. And different people touch a different part of the elephant, have a different idea about how the elephant looks like. If you sample one data point, if you only from this angle, you see part of the truth. In science, that's the same thing. So sometimes we have a big argument in science, like A scientists say this is A, B scientists say you're wrong, it's B. Just think about the story. They are probably, if both of them are right, then they're probably looking at the same thing from different directions. Contrast here is use a specific short sequence information from one particular place. This is like a corpus, or you want to look at the overall differences globally from the whole genome. Of course, I want to look at the global view. We want to have visions. So start from our school year, everybody said we have to have visions. You don't really know because our vision are covered by the buildings. And in the city, you really can't see very far. And what does people really talk about? What our teacher teach us? And those are really very difficult for young kids to understand. I don't think I understand it all. But some of us might, if you are smarter than me, you probably already understand what your teacher really say. But here he's saying, if I tell you, I want to see things from a single point, or I want to see things in global view, I think majority of us say, of course you want to see things overall, you want to get the whole idea. And that's why the DNA hybridization was a very important technique. And a lot of people do that and just want to see, I want to see the overall differences. I want to get everything. I might not know the ATGC in order, but I want to know how much similarity in the entire genome. So that's the idea for the DNA hybridization. And then that's the genomics. You'll know that. So after genomics, things are happened very quick. The previous year was 1984. We have DNA hybridization, and in 80s, that's the technique a lot of people use. And another thing I want to say is, occasionally we hear students, or especially graduate students, think about this technique is out of date. Well, based on when they discovered, it's probably out of date. For example, DNA sequence, right? DNA sequence was invented in 1977. Oh, it's very old. But we're still using it, right? We use a different machine, and we use a very different method, but we still use the same principle Sanger used to sequence today's molecule. And there's some variation about it, but more or less they are very similar. At this, a majority of machines still use the same method or similar method to read DNA sequence. But on the other hand, people say, oh, electrophoresis, who does that? And it's an old technique. Think again. Every technique has its own history and has its advantage. If it's convenient for you to use it, if it can help you to understand the question well, why not? If you can use a method developed in the 30s and get a very good new discovery and spend very little money, that is very good, right? So it's hard to compare which method is new or old. And sometimes a combination of methods can give you a big improvement. So you don't have to invent new parts of new tools, but you can make advance in our life. And that's the same thing organisms use. And they don't really think about it to design it. But if things happen, it works, and it stay in the population, and we'll see it. And that's how evolution happened. So this is something we really have to think about. So nothing I'll talk about after we have the genome. The reason is that's just too much information. And for the history part, I only talk about to here because all the new methods, all the data we'll talk about through the class. This is just a very early history about the molecular evolution. So actually the title was given, I believe it's given by Professor Wensheng Li. It's a pre-DNA. So we talked very little DNA at the end. So we didn't talk about the polymerase chain reaction technology. So there's a lot of technology made the data available quickly. And we haven't really talked about what happened in the past 5 years, why genome, we get the genome much easier than, say, 10 years earlier. So, but I hope you will enjoy the class, and we'll talk more about that. So all the terminologies, basic terminology, we'll do that in the next class when we meet.